Start.
Good morning. So I will be talking about this article with the title Durable Oral Biofilm Resistance for 3D Printed Dental Based Polymers Containing Zwitterionic Materials. So firstly, to know, this was published in the journal International Journal of Molecular Science on 2021. With the impact factor of 6.2. The aim and novelty is as follows. The polymethyl methacrylate, which is PMMA, it is the base polymer which is routinely used in dentistry due to its various advantages. Recent advantage enables the ability of 3D printing the PMMA as SLA printable dental material. So microbial colonization on denture surfaces remains the major drawback, causing the spread of infection. So here they added Zwitterion molecules. Zwitterions are molecules with highly negative and highly charged cationic and anionic parts within the same molecule. So they have a higher dipole moment, and overall it is overall neutral, but with strong electrostatic interactions. So, for example, 2-meth-acrylyl oxyethyl phosphoryl chlorine MPC and sulfobetane methacrylate SB. So Zwitterions have protein repellent, anti-fouling and antibacterial effects, making them useful material to prevent microbial colonization by promoting surface protein detachment. The in vitro study aims to evaluate the effect of MPC and SB between PMMA based dental devices and oral salivary biofilm, along with the clinical features, deterioration and durability of 3D printed PMMA. The objective of this study was to study the difference between PMMA with and without MPC and SB without the Zwitterions. In terms of physical and mechanical properties, oral salivary biofilm resistance, and mentioned properties following hydrothermal fatigue. The same properties were tested before and after hydrothermal fatigue. So the methods: preparation of Zwitterions incorporated in 3D printed PMMA. MPC and SB Zwitterions powder 3% by weight in distilled water. It was mixed at 3500 rpm for 2 minutes and then mixed with PMMA. 3D printed blocks were made using light processing 3D printer, followed by 10 minutes post-process in UV oven and polished. 3 groups were made: PMC PMMA, SB PMMA, and a PMMA without Zwitterions, which was as a control group. So physical and mechanical properties were tested. Firstly, wettability. The measured contact angle between the contact angle was measured using goinometer and by adding water droplet on the disc-shaped sample. Flexural strength and elastic modulus were calculated using ISO standards using universal testing machine. Viscous hardness was measured using tester. 2.94 Newton load was applied for 30 seconds. For protein absorption, samples were immersed in 1 hour PBS, followed by 1 hour in protein solution and PBS rinse at room temperature, followed by humid sterile incubation for 4 hours at 37 degrees Celsius. Protein adhesion determined by using biofilm-related acid modification. Bacterial adhesion. The bacterial adhesion was tested for using Streptococcus mutans, Staphylococcus aureus, Klebsiella obscura, and Klebsiella pneumoniae, as they are early colonizers of biofilm on PMMA according to the previous studies. Cultured bacterial suspension was added to sterilized samples and cultured for 24 hours and then washed to remove the unattached bacteria and then stained and observed under the confocal microscope. Pre and post thermocycling assessment was done for S mutants by immersion of the sample in the bacterial suspension. Solidation of the sample was done and bacterial suspension was serially diluted, spread on BHI, that is brain heart infusion agar, and intubated for 24 hours for colony counting. So, human salivary oral biofilm on 3D printed PMMA. Saliva samples from 6 individuals were taken and cultured for 48 hours, then stained and analyzed by confocal laser microscope. Biofilm thickness and biomass was measured using ComStat plugin and ImageJ software. Dimensional accuracy of PMMA. It was the image acquired from three shape E3 scanner was used to 3D print the denture based models according to the 3 groups. Dimensional differences between original scan data and treated samples were compared using a 3D morphometric program. So hydrothermal fatigue, all these tests were done and then in the normal temperature, and after that they were repeated. All these tests were repeated after thermocycling the samples between 5 and 55 degrees Celsius with a dwell time of 45 seconds for 850 cycles corresponding to 1 month. So statistical analysis, it was performed by using data from the least 3 independent experiments using statistical software. The results were analyzed by ANOVA and post-hoc Turkey test. The P, which was less than 0.05, was considered to be statistically significant. So the results, addition of either Zwitterion into PMMA, either MPC or SB, resulted in significant reduction in the contact angle compared to that of the control. So after thermocycling, the PMMC group had a lower contact angle. Both the flexural strength and elastic modulus decreased insignificantly, but significantly reduced in viscous hardness following the addition of Zwitterions. No difference in the results were seen after thermocycling. Significant reduction in the protein absorption was seen compared to the control group, with no significant difference in both the Zwitterion groups. Sample shows more growth for all bacteria in the control against the Zwitterion groups. No statistical difference was seen between both the Zwitterion groups. There was a significant reduction in S mutants colony forming units found for both Zwitterion groups, with no statistical difference among the group, which was maintained even after thermocycling. The biofilm formation was abundant in the PMMA when compared to the Zwitterion incorporated PMMA. Increased biofilm formation post thermocycling was seen in Zwitterion incorporated PMMA. Significant reduction in the biofilm thickness and biomass was seen in the PMC-SB integrated PMMA. The control sample had a 1.4 times thickness of the biofilm compared to the MPC PMMA, which means the Zwitterion induced PMMA had less thickness compared to that of only the PMMA without the Zwitterion. Also, tolerable dimension changes were noted with no statistical difference between all the three groups after thermocycling. So there was no dimensional changes significantly which was noticed in the three groups. So the strength and limitations for this study can be as follows. The strengths are multi-dimensional testing of the properties and of the properties of the material were done. Prevention of biofilm formation and adhesion was tested. Incorporation of hydrothermal fatigue testing by thermocycling, it gave a better results and replicated the natural oral environment. The novel anti-fouling and anti-fouling materials for biofilm resistance was found. Maintenance of stable dimensional accuracy and compliance with ISO for dental applications. Limitations: lack of in vivo validation, limited bacterial species, reduced textural strength and hardness, decreasing long-term durability, short duration of 850 cycles in thermocycler. It may not reflect the actual clinical scenario. The use of mixed human saliva for biofilm test induces variability as the composition of saliva differs among each individual. Lastly, the potential for long-term effects of MPC and SB on the oral tissues were not evaluated. So, basically the conclusion is that Zwitterion incorporation in the denture acrylic can reduce the biofilm formation and prevent the microbial growth. But in this study, they have not replicated the experiment in the humans, so that can be done for a better result. Thank you.